Hello, everyone. I would like to welcome you to today's webinar on Rapid Evidence-Based Reporting of Oncology NGS Tests at Scale, which will be presented by Dr. Beate Litzenburger and Dr. Sheryl Elkin. My name is Mary Ann. I'm the Global Campaign Manager for QIAGEN Digital Insights, and I'll be the host and moderator for today. I'd like to introduce our speakers for today, Dr. Beate Litzenburger and Dr. Sheryl Elkin. Beate is the Global Product Director of Oncology for Clinical Decision Support Software and Interpretation Services at QIAGEN. Prior to joining the QIAGEN team, Dr. Litzenburger led clinical decision support and the interpretation of patient cases for several next-generation sequencing protocols at the MD Anderson Cancer Center. In this role, Dr. Litzenburger was responsible for providing alteration interpretation, drug, and clinical trial options to clinicians. Sheryl is the Chief Scientific Officer at N of One, a QIAGEN company. An early member of the N of One team, Dr. Elkin has led the interpretation of thousands of patient cases, establishing a rigorous process for the analysis of scientific and clinical evidence and presentation of molecular and clinical evidence to physicians to help guide their therapeutic decisions. Dr. Elkin has taken a lead role in the development of the N of One clinical interpretation methodology to support clinicians in identifying therapeutic strategies specific to each patient. So again, thank you for joining us today, and I will now hand the presentation over to Beate and Sheryl.
Thank you so much for the introduction. QIAGEN Digital Insights offers a variety of products and services to meet your needs to call and interpret variants from NGS assays. So let's focus here only on the tertiary analysis solutions. For molecular diagnostic labs performing variant interpretation and reporting in-house, QCI Interpret is a clinical decision support software that can be integrated in your existing workflows and enables fast turnaround times, high confidence reporting, and test menu expansion for oncology and hereditary diseases. Now, QCI Precision Insights is a professional clinical interpretation service that translates molecular data specific to each patient into clinical insights and therapeutic options. QCI Precision Insights is focused on interpreting tumor profiles. Powered by a world-class team of molecular biologists, and oncologists, QCI Precision Insights delivers expert assembled reports with detailed assessments of variants, written summaries of evidence with references, treatments, and geographically matched clinical trials. The professional interpretation service allows labs to scale without extra staffing and reduces the test turnaround time by about 85 to 90%. Now, all our tertiary products can be customized towards your needs and help you to scale using API integration, workflow customization, and automation, LIMS integration, and variant filtering optimization. Our professional services also develop report templates that allow you to differentiate yourself for brand recognition. Competing to offer a comprehensive genomic profiling service for tumor samples is challenging, especially as panels increase in size and complexity. That means that we are more than ever challenged to classify even more variants. At the same time, oncology drug development advances and an increasing number of treatment decisions require evidence and options beyond the current guidelines, which need to be guided by the appropriate level of clinical evidence from the literature. The expectations for thoroughness of clinical reports are high which requires a more than ever comprehensive variant and disease-specific research. And of course, all this increased effort needs to be done while maintaining rapid turnaround times and achieving high accuracy. Therefore, there is a need to reduce the time-consuming task of researching and writing variant and disease-specific evidence summaries. In addition, there's also a need to be confident in the accuracy of the variant classification by consulting an expert second opinion, and to create an accurate, content-rich report ready to be sent to oncologists. We created QCI Interpret 1 by combining the flexible and automatable QCI software powered by superior structured content in the QIAGEN knowledge base with the trusted interpretation service of NF1, a world-leading provider of somatic variant interpretation. Now, QCI Interpret 1 allows labs to confidently interpret variants with disease-specific oncologist-reviewed content, as well as access an expert second opinion for increased confidence in accurate variant classification. Keysight Interpret 1 allows labs to meet their turnaround times without the time-consuming step of researching and writing variant and disease-specific evidence summaries. And with easy editorial control over the report content and layout, you can deliver professional reports directly to physicians to help inform clinical decision-making. Now, the oncologist-ready reports provide a comprehensive overview of the patient's molecular profile. Expert variant and disease-specific summaries are provided for biologically and clinically relevant variants. The first page of the report not only provides an overview of potentially relevant treatments, but also provides a note to the oncologist of any interaction, meaning if co-occurring mutations may impact prognostic, diagnostic relevance, or may affect therapeutic outcomes. Oncologists will also receive a note if guideline information is available, which then they can review on a subsequent page of the report. So at a high level, how do you generate those comprehensive reports using Keysight Interpret 1? So data files are uploaded and contextualized minimally with a patient's disease type to allow for accurate display of disease-specific content. In the first step, variants are prioritized based on the most clinically relevant variants. In the subsequent step, you can classify the variants and report treatment and trials. The last step, the report building step, allows you to retrieve ready-to-use variant and disease-specific summaries without the time-consuming step of writing those by yourself. In addition, our experts provide a second opinion on the variant classification to increase the confidence in the accuracy of your classification. And then the final reports can be generated in form of a PDF or can be downloaded or integrated in the LIMS using XML. Keysight Interpret 1 is therefore powered by the aggregated knowledge from the QIAGEN knowledge base, combined with a trusted professional interpretation service from NF1, which has been a QIAGEN company now for over a year. Now, Keysight Interpret 1 is built on a flexible platform, which allows you to access many turnkey workflows for a variety of commercial and off-the-shelf panels, but also allows you to configure the workflows according to your needs. In addition, you have the flexibility to manually interact with the software in a user interface, or partially or completely automate the workflow to reduce the hands-on time and therefore increase the turnaround time. Keysight Interpret 1 takes advantage of more than 20 years of expert curation of genomic and cancer-related literature aggregated in the QIAGEN knowledge base, combined with more than 10 years of accumulated knowledge in providing professional interpretation services for somatic variant interpretations, and thus empowers your lab with the world's largest manually curated knowledge of biological and clinical variant and disease-specific information. Now, I want to first focus on the QIAGEN knowledge base before Dr. Sheryl Elkin will take over and discuss and introduce the curation and interpretation service. So, what differentiates the QIAGEN knowledge base from other databases? It basically transforms unstructured data from the literature into machine-readable units to allow computing of content for every patient case. The knowledge base structure allows us to aggregate manually curated scientific literature and professional guidelines and model the data with semantic consistency. The knowledge base allows us to capture biological, phenotypic, therapeutic, and outcome data for subsequent transformation into clinically relevant information for each patient case. So how does QIAGEN transform unstructured data into computable units? So this is accomplished through a unique knowledge-based model and curation process. The expert curators, which are Ph.D. and M.D. levels, read the scientific literature, including the figures, tables, supplementary information, and use rigid curation protocols and curation tools to add each relationship, such as an alteration, clinical effect, drug and disease association, into a structured format in the knowledge base. Now, we can use these relationships and units to compute the pathogenicity and actionability for each variant and return variant and disease-specific information for each case. In addition, we use natural language generation processing to return in a human-readable sentence the essence of an experiment, for example, a functional study or a clinical trial result from a paper. Now, how often is the data in the QIAGEN knowledge base updated? To ensure an up-to-date knowledge base, the content is updated mostly on a weekly basis, which allows us to cope with the increasing number of variant-specific literature, the changing information from labels, trials, guidelines, and an increasing accumulation of expertly written molecular function summaries powered by NF1 and changing disease relationships. Keysight Interpret 1 displays information from a variety set of different databases. Those databases allow you to rapidly assess the frequency of this alteration of individuals from the general population, as well as understand the somatic frequency of this alteration and context of this diagnosis. Keysight Interpret 1 allows you to retrieve or review records for that exact variant from other laboratories, and thus saving you time of doing the individual queries manually. To note, Keysight Interpret 1 supports two gene models. That's the RAF-seq gene model and ensemble gene model. Now, why do I spend so much time providing insights into the QIAGEN knowledge base? It's because the machine-readable data allows computing of content for each patient case. So what does that mean for variant interpretation? First, Keysight Interpret 1 can instantly prioritize clinically relevant variants from any assay. Second, Keysight Interpret 1 is able to provide for every variant across a whole genome in any cancer type the computed ACMG and AMP-based classification, the molecular function, incidence and disease, if hereditary clinical cases are available, treatments, variant and disease-specific outcome studies, and open and recruiting clinical trials. So let's dive into the QCI Interpret 1 application. So how is this information from the QIAGEN knowledge base displayed in QCI Interpret 1? This is the variant list of an NGS result of a breast carcinoma patient sample. Each row represents an alteration detected in the tumor tissue. The different colors, such as red, pink, or gray, display the QCI computed pathogenicity of an alteration. And in the bottom center indicates the QCI computed actionability classification. The smaller batches to the right, such as the S or R, provide an initial clue what information is available for this variant. For example, if variant is sensitive or resistant to therapies. This view basically provides a broad overview of the patient's molecular profile and allows flexible filtering to prioritize the alterations that would require a deeper review. Now, the variant list page allows you to also identify variants that have been previously observed by experts from N-of-1, where there is an indication of an E icon on the tile of a variant. N-of-1 has previously observed and expertly interpreted over 170,000 variants. And using the combined knowledge from the QIAGEN knowledge base and the expert allows you to confidently distinguish variants of uncertain significance from those that may have clinical relevance. So, just as an example, clicking on the functional icon opens the reported functional impact section, which provides you with an expertly written summary of the molecular function backed up with references. In addition, you have access to the curated references from the QIAGEN knowledge base and computed biochemical impact from various prediction tools. And the combined knowledge allows you to confidently interpret the functional impact and relevance of this mutation. Now, in a different example, alternatively, the combined knowledge allows you to confidently distinguish variants of uncertain significance from those that may have clinical relevance. So in this example here, QCI Interpret 1 does not display literature evidence from the QIAGEN knowledge base about this variant. Now, on top of that, N-of-1 experts did a targeted curation of this variant and specifically researched the literature and database sources and determined its unknown impact. Now, for each variant that is detected in the patient's profile, QCI Interpret 1 provides a one-stop shop organized in several expandable sections for you to review detailed information about the alteration and context of the user-submitted phenotype. In the interest of time, I'm not able to dive into the details of each section, but I want to highlight a few important sections. The laboratory observation section is a quick and transparent way to review and track the history of the variant classification across several different phenotypes. And the effect on protein section allows you to quickly understand the location of the mutation in a gene and if it falls into an important domain highlighted here in yellow. It also allows you to see if the variant falls into a region where typically pathogenic mutations are observed. This view also provides an idea if mostly gain-of-function mutations or loss-of-function mutations are observed in this gene. Further highlighted in this red box are the PIK3CA variants found in this patient. In this case, you can see that this patient contains a PIK3CA double mutation. The observation of patient has a single or double mutation in a gene may be of clinical relevance, as double mutations can lead to an increased or decreased drug sensitivity. Now, Keysight Interpret 1 provides in the variant details section front and center an assessment section, which displays the computed actionability and pathogenicity criteria. The actionability is based on AMP guidelines. The pathogenicity is based on the ACMG guidelines. So here I want to focus on the actionability for this variant. All tiers that are applicable will be shown to transparently indicate the actionability options for this alteration. For each tier, the underlying clinical evidence can be viewed, which provides transparency in the sources that support each tier. One thing to note is that the user still has full control of adjusting the actionability criteria by removing or adding a criterion, or just manually adjusting the final assessment of the alteration. In this section, the user has the ability to report out the pathogenicity and actionability for each alteration. Now, within the treatment section of the variant details page, Keysight Interpret 1 shows pre-selected treatments for the draft report, which is, of course, adjustable. For each drug response, a level of evidence is provided in context of the disease, and for transparency, references are provided. Now, the last step is the report building step that allows you to generate comprehensive reports in minutes without the time-consuming step to write interpretive comments by yourself. In the review and report page, Keysight Interpret 1 displays all the variants that should be reported and also indicates in the expert interpretation column which variants will receive variant and disease-specific interpretation. And this is done with a simple click on Submit for Expert Interpretation. Now, in this moment, I'm handing over to my colleague, Dr. Elkin, who will describe in depth the expert interpretation component of QCI Interpret 1.
Thank you, Dr. Litzenburger, for your excellent presentation and kind introduction. The expert interpretation component of QCI Interpret 1 is driven by QCI Precision Insights, the former N of 1. QCI Precision Insights provides human analysis on top of the computed variant classifications provided by QCI Interpret, allowing for detailed interpretation of gray areas in complex scientific situations. Precision Insights also triages and summarizes content for lab director review to assist in report writing, helping to further shorten turnaround times. So who is N of 1? N of 1 was founded in 2008, and we have grown and evolved with the field of precision medicine. Back at the beginning, we worked directly with physicians and their patients to help them to identify the most appropriate molecular tests for their tumors, get the tests done, and then explain the results. Through the process of working with individual patients and physicians, N of 1 established a knowledge base of biomarkers, genes and variants, in the context of different cancer types linked with drugs that had emerging or established evidence that they could target those biomarkers. In 2012, we began working with large hospital systems and commercial laboratories, taking in the results of patient molecular tests and providing reports. And by 2016, we had partnered with over 30 clinical laboratories to provide clinical interpretation. In 2019, we joined QIAGEN, having generated reports for over 100,000 patient cases with a deep knowledge base covering over 1,800 genes and over 100,000 unique variants. These numbers continue to grow every day as we have now processed over 200,000 cases. In 2020, our professional interpretation service has expanded clinical variant interpretation services to Europe and have now integrated with QCI Interpret to create QCI Interpret 1, an end-to-end clinical interpretation product that takes the user from VCF to final report. QCI Interpret 1 accesses the accumulated knowledge of N of 1, which has assembled over 12 years of manual expert expert clinical interpretation by full-time, dedicated Ph.D. scientists. The scientists undergo a rigorous training process, including defined practices for analysis and interpretation of scientific studies. QCI Interpret 1 provides report-ready variant and disease-specific interpretation summaries on all kinds of biomarkers, including SNVs, indels, fusions, and CNVs, as well as support for microsatellite instability and tumor mutation burden. The variants are described specifically in terms of their molecular impact and are classified according to the AMP guidelines published in 2017. Variant and disease-specific clinical evidence covering potential therapeutic options as well as prognostic and diagnostic significance where applicable is reviewed and approved by a team of practicing medical oncologists. This team of oncologists practices across oncology subspecialties and have been collaborating with the N of 1 team for many years. The QCI Interpret 1 interpretation also includes summaries of disease and gene-specific relevant treatment guidelines, as well as analysis of multivariant interactions between variants in the same gene or between different genes. These interactions are selected based on the level of evidence and pre-curated based on the literature, and detection and analysis of the interactions can be fully automated and reported. As of today, over 200,000 cases have been interpreted, with over 170,000 unique somatic variants across over 1,000 genes and 1,000 cancer subtypes. The expert-written interpretation summaries within QCI Interpret 1 are configurable. The information is highly structured, and users can configure which sections they would like to view within the user interface and in the final report. We have pre-configured 3 options: a complete option, which contains the deepest and richest information, a concise option, which contains only the highest level summary information, and an intermediate version. The configurations are also flexible in the event that the pre-configured options do not meet a user's needs. So how does this information appear in the user interface? When Beate left off on the review and report page, she had clicked the Submit for Expert Interpretation button. Typically, within a few minutes, this page is then populated with the variant and disease-specific expert interpretations. In the overall interpretation section shown here, you will see information that applies to the case as a whole, summaries of any applicable treatment guidelines, and reference descriptions of any variant interactions. Below, you will see the list of reportable variants, and the expert interpretation column has now been populated with colored squares, with green indicating that the experts agreed with QCI and the yellow indicating a disagreement. If you click on the triangle next to the gene, you can open the expert interpretations. Here we have opened the PIK3CA variant, and you can see we are showing an example of the rich content at the deepest level of detail. The content is highly standardized and structured and always in the same order, so that the user always knows where to look for a given piece of information. For example, if they want to know if the variant is associated with sensitivity to any drugs, they would look down to the drug sensitivity section. If they wanted to know the specific molecular impact of the mutation on the gene, they would look up to the molecular function section. Sections for diagnostic and prognostic significance are also included, as you can see here, but they are seldom populated in solid tumor cases, as the prognostic and diagnostic impact of molecular alterations is more well studied in heme and neuro-oncology. Still, all sections are included so that the user always knows where to look. Additionally, every sentence is referenced so that if a user does want to review the primary evidence, it is provided. The interpretation is fully transparent to the evidence that was used to build it. The content from the expert interpretation can be used as a second set of eyes to help verify and give confidence in the variant classification and interpretations. For example, the variant shown here is a JAK3 truncation that was computed as a Tier 3 unknown significance variant. In this case, the expert interpretation is in agreement with the QCI computed classification. the JAK3 truncation is likely deleterious to the protein, but because activation of JAK3 has been implicated in cancer, the relevance to cancer of a JAK3 truncation is unknown. The expert interpretation in this case supports the computed classification. I would also like to point out the edit button here. The users have editorial control to refine the content for the report in the event that they would like to add or remove a piece of information, change wording, or otherwise revise the content. These changes can be saved and will be easily accessible for future cases. After the lab director has reviewed all the variants that they wish to review and made any desired changes to content or classification, they are ready to preview and sign out the report. The buttons at the top of this page allow flexibility to assign reports within different users and different roles within an organization, so that if a variant scientist is working on a case, for example, they can assign it to another scientist or to a lab director for sign-out. The user can also click the Preview Report button to generate a PDF report. And here is an example of the draft report. The version you see here is the QCI Interpret 1 report template, but it can be customized for individual laboratories, incorporating logos and making other changes. Additionally, the content can be exported via XML and uploaded into a laboratory's own report template or LIMS, allowing the laboratory ultimate flexibility to build its own report and establish a brand. At the top of the report, you see an overall comment, which can be composed in the user interface. If no overall comment is supplied, this section is not included in the draft report. The variants are presented on the summary page according to the guidance in the AMP guidelines. Tier 1 contains variants of strong clinical significance, variants that are associated with therapies approved within the specific indication. Also shown here are therapies with evidence of resistance, as well as clinical trials. Tier 2 contains variants of potential clinical significance, and also shown on this page are variants of biological significance, clearly pathogenic variants, and likely drivers of cancer, but with no available targeted therapies or trials, and without significant prognostic or diagnostic relevance. The front page also has pointers that inform the user to information about interactions and guidelines, as well as a table of contents. Getting into the body of the report, there is a guidelines section that appears when applicable. This contains a summary of disease-specific guidelines that are relevant to the detected biomarkers, in this case, ERB2 and PIK3CA in breast cancer. The interactions section summarizes interactions between variants. As described earlier, the interactions are researched by our team of Ph.D. scientists and reviewed and confirmed by oncologists. They must reach a defined level of evidence for inclusion. Interactions can affect drug sensitivity and resistance. They can also modulate the prognostic implications, and combinatorial variants can be used to inform diagnosis. In this case, there is some conflicting evidence. PIK3CA and ERB2 alterations do not often co-occur, and PIK3CA mutation may be associated with resistance to ERB2 targeted therapy, but the evidence is unclear. The ERB2 targeted therapies are still included in the report. The information here is provided so that the physician is aware of a potential cause of resistance that they may observe. Treatment options are shown on this page as well. Users have the options to review and select from the proposed therapies directly or to automatically report therapies using a predefined reporting policy as described earlier by Dr. Litzenburger. The text that's shown here in the report describes the information that is contained on the drug label. Clinical trials can also be manually or automatically selected and are shown here organized by the phase of the clinical trial. The next page shows the detailed clinical interpretation that has been provided through the Expert Interpretation Service. In this slide, we are again showing the most detailed, rich information possible. As I mentioned earlier, the amount of content displayed is configurable. The more concise content is supported by the rich content seen here. You always know that even if you are only reading the concise content, the detailed research has been done by the PhDs on the N of 1 team. The information is highly structured and presented in increasing levels of detail. The information shown first at the top is a high-level summary. What kind of variant is this? An activating mutation? What is the role of variants in this gene in cancer? Are there any therapies available to target this gene? The disease summary gives the most important gene about this gene in this disease. The next sections get into the biological relevance of the alteration. The molecular function section describes the impact of this specific variant on the protein, along with all the supporting references. What does this variant do? Is it activating? Is it inactivating? What is the evidence? Are there biochemical studies demonstrating kinase activation, or does this mutation lead to transformation of cancer cells or tumors in mice? Next, what is the incidence of alterations in this gene in this cancer type? And finally, role in disease. What role does this gene play in the development of cancer in general, or in this cancer type specifically? The next sections describe the clinical relevance of the alteration. Is there diagnostic or prognostic significance for this mutation? Does the mutation predict sensitivity to any drugs, resistance to any drugs? And then finally, the most detailed information, the clinical evidence section. This section provides detailed data to support the more summary level information that precedes it. The information summarizes data from individual clinical trials. It is specific to the cancer type and organized by the phase of the information from which the data is derived, or the phase of the clinical trial from which the data is derived. This information tends to be most often used by academic laboratories and molecular tumor boards. It allows them to see the specifics of the data without needing to take the time to do the research, identify, and read the papers. The information shown is, we will show the highest level information for any given drug or any given therapeutic approach, so that if a phase 3 study is shown in summary here, the same drug information would not be shown from an earlier clinical trial unless it's in a different clinical context or a different drug combination, and therefore limiting the extent of this very deep and detailed data. This is an example of a concise version of the report. As explained earlier, the content sections included are flexible, but this is one example of the high-level information that might be most critical without including all of the supporting details, which again, the scientific team has done the research to make the summary conclusions. To summarize, QCI Interpret 1 brings together the knowledge and expertise from the QIAGEN knowledge base and over a decade of N of 1 expert interpretation to create an end-to-end solution that is unparalleled in the field. QCI Interpret 1 combines flexible software with state-of-the-art interpretation that allows users to confidently interpret variants using the power of computed classifications and a second set of expert eyes. QCI Interpret 1 helps laboratories to accelerate turnaround time by providing expert-curated, oncologist-reviewed written summaries that can be directly added to a report or edited to reflect the preference of the laboratory. In the end, the user has full editorial control over the classifications, content, drugs, and clinical trials, as well as the design of the report that can be configured to fit the needs and brand of the lab, all of which will help a laboratory to differentiate and succeed in the market. And with that, we thank you for your attention. If you would like to request a demo, please go to the displayed website or Google QCI Interpret 1 to access the website for requesting a demo. And with that, we would be happy to take any questions that you may have. Thank you very much.
Thank you, Sheryl and Beate, for that presentation. So now we're going to have some time to go over any questions that you have. Moving into the questions, the first one is, you talked about the update frequency of the QIAGEN knowledge base. how do you update the interpretive comments?
Hi, so I'd be happy to take that question. The interpretive comments, the expert interpretation, are coming from the N of 1 team, and that it's a live knowledge base that's being updated on a regular basis. Everything in the knowledge base has an expiration date, so to speak, so things are updated at least annually, but every different piece of information has its own amount of time that it's allowed to expire. So we're making sure that we're reviewing the information regularly and preloading new information. Additionally, new drug approvals are incorporated within a business day, and updates to guidelines are also incorporated within 1 to 2 business days.
Okay, thank you, Sheryl. Our next question is, how are the oncologists involved in the process of validating the clinical evidence in the interpretation summaries?
Sure, so I can take that one as well. We have a team of 9 oncologists that have been working with us for many years. They span the clinical subspecialties, from heme to neuro-oncology and multiple different types of solid tumor focuses, including thoracic and breast, GI. And they regularly review the annotations that our scientists are bringing into the knowledge base. So any piece of information that's summarizing the literature that has to do with anything clinically related, so a clinical impact of an alteration or a review of a clinical trial, that would be submitted to our oncologists for review. And we've developed a very efficient system to alert them that there's something for them to review, and they're reviewing within our system on a daily basis. They're all practicing oncologists. Most of them also have active research programs. And they also consult with us on associations of drugs and clinical trials with particular biomarkers and other scientific topics that we are looking for extra clinical input. So they lend a real clinical eye to the information that we're providing.
Okay, thank you. We've had a couple questions about input files. So what kind of variants can QCI Interpret 1 process and interpret? And what file types can be inputted into the informatics system?
Yeah, I'm happy to comment on that. So Keysight Interpret 1 is the tertiary analysis solution. So if we go straight into Keysight Interpret 1, the standard format is a BCF format that follows the standard 4.x, I would say, formats. Now, there are outputs from other pipelines, secondary analysis pipelines, that output several different formats. One specifically I have in my mind is the TSO 500 platform. They output several different formats outside of the BCF format, and we're able to intake that as well. Now, there are some platforms that do have very unconventional output files. So we do have some methods that allows us to upload the data via an XML into the system, or we do have in the user interface also a way to upload this, in this case manually, but with the XML is an automated way. The other question was, what are the alteration types that are supported? So the output files from a secondary analysis typically get into the copy numbers, diffusions, and small variants, and so all of them are supported. We do support anything that we can encode in a structural variant files, insertions, larger deletions, smaller deletions, insertions, inversions, and of course the copy numbers as well. So those are the structural variants, the small variants, SNVs and DELTs. We do support other formats as well. As you're calling TMV or MSI, you can upload it as well. into the pipeline, and we interpret that. So there is another question around if we can incorporate FAST-Q files. So it depends on your pipeline. So we can run the second analysis pipeline and automatically upload into our system as well. So if you wish to use a FAST-Q all the way to report, that's possible as well.
Great, thank you, Beate. The next question that we have is, how long will it take for the professional interpretation services to provide me with the interpretive comments?
Sure, I can take that one as well. So we have two different levels of service. So the first one would be the pre-curated version, and that would return expert comments within just a couple of minutes, 1 to 2 minutes. Usually, and that is drawing the information off of our database, based on over 200,000 curated cases and variants, and pre-curated information as well. So whatever is currently present in the knowledge base would be pulled in to the QCI user interface. We also, and in that case, if there were a variant that were novel that hadn't been covered by the N of 1 team, then it would be returned as variant not curated by N of 1 with the caveats in the appropriate places, but returning gene and disease level content as available. We also have a curation service option where the team will curate every piece of information for the report. So every novel variant, filling in information on the gene and disease level as necessary, doing any required updates, and that we promise within up to 3 days, but usually on average those are turned around also within on average about a half day.
Okay, thank you, Sheryl. Next we have, when you were showing the sample report, the slides were on a solid tumor sample. Does QCI Interpret 1 process heme or neuro-oncology samples?
Yes, we do. Both the QIAGEN knowledge base and the N of 1 team expert interpretation knowledge base both cover solid tumors, heme, and neuro-oncology as well. And our oncology experts span those subspecialties as well. So we have input from clinical hematologists, oncologists, and a neuro-oncologist that weigh in, again, from that clinical perspective.
Okay. I think we have one more question left. Again, if anyone does have any additional questions, please use the Ask Questions panel on the left side of your screen to submit them. So the last question that we have for now is, does QCI 1 cover TMB, MSI, fusions, and CNV?
Yeah, I think I commented on that before. Is that, yes, we do cover TMB, MSI, fusions, and copy numbers. So again, depends on the upload files. We would display the information. I want to comment on one thing, which is the fusions. It is important to take into consideration the fusion breakpoints. So we do display that the fusion breakpoints that are encoded in your files, and display that information. Then when and at the same time, so when you're consulting our experts for interpretation, and they take those breakpoints into consideration for interpretation, and that's important to understand if a kinase domain is cut off, or if certain important functional domains are fused together that create a potential oncogenic fusion or potential kinase dead fusion. That's important for then targeting these particular fusions for appropriate drugs or clinical trials.
Okay, great. Thank you, Beate. So that looks like we've reached the end of our questions. So I'd like to thank our speakers for today, Dr. Beate Litzenburger and Dr. Sheryl Elkin. I also want to thank the audience for attending, for your attention, and also for submitting the questions. So this concludes today's session on rapid evidence-based reporting of oncology NGS tests at scale, and we look forward to hosting you in a future webinar. So thank you, everyone, and I hope you have a good rest of your day.